cardiovascular system, but we're starting off with blood vessels in general, the arteries, the capillaries, and the veins. Here are all the understandings I'm about to cover and review. Make note of the last one because we don't identify blood vessels by the type of blood they're carrying anymore, but rather the structure of their walls, as there are two exceptions to the deoxygenated and oxygenated blood rule, which would be the pulmonary artery and the pulmonary vein. So first off, we do have arteries. First understanding is that arteries convey blood at high pressure from the ventricles to the tissues of the body, the ventricles being located in the heart. And arteries transport blood from the heart that is on its way to the capillaries. The second understanding would be that arteries have muscle cells and elastic fibers in their wall. Arteries do have a relatively thick, smooth muscle layer that is used by the autonomic nervous system to change the inside diameter or the lumen of the blood vessels, which means that they are stretchy, and this is furthered by the elastic fibers that assist in the maintenance of a relatively high blood pressure achieved by the contractions of the ventricles in the heart. Notice in the diagram its external elastic part and the elastic membrane. So this understanding that the muscle and elastic fibers assist in maintaining blood pressure between the pump cycles of the heart is that in the diagrams you can see that elastic fibers stretch and let the blood vessel accommodate for increased pressure caused by blood being pumped into the artery. But also when this contraction is over, these elastic fibers provide another source of pressure by closing back down when they return to their original position. Second off, we do have capillaries. The understanding is that blood flows through capillaries. Capillaries have permeable walls that allow for the exchange of materials between cells and the tissues and blood in the capillary. Blood enters a capillary bed through an arteriole, which is the smallest of the arteries, and then the blood cells flow into small, single-cell thick capillaries, which allow for material exchange because the walls of the capillary are so thin, one cell thick. Lots of blood pressure is lost when blood cells enter the capillary, and chemical/material exchange between blood and tissue has to occur in the capillary because artery and vein walls are too thick to allow for an efficient diffusion of molecules between the cells. Lastly, we do have veins, which return blood to the heart. The understandings would be that veins collect blood at a low pressure from the tissues of the body, or the capillaries, and return it to the atria of the heart. Valves in the veins and heart ensure circulation of blood by preventing backflow. So, since the blood is very low pressure when it's going back to the heart, the blood flow is much slower and is also usually fighting against the force of gravity as the heart is above most of your body. So as you can see in the diagram, these veins have internal valves which help slow moving blood travel consistently towards the heart, and these valves also prevent backflow so your blood doesn't just all clog in one area. Think about these valves being kind of vertical pumps.
The understanding for this section is the recognition of the chambers and valves of the heart and blood vessels connected to it in dissected hearts or in diagrams of heart structure. The human heart is designed as a pair of side-by-side pumps. Each side of the heart has a collection chamber for the blood that moves in slowly from the veins. The atria is the thin-walled muscular chambers at the top of the heart, on the left and right side of the heart. The ventricles are the thick-walled muscular pumps at the bottom of the heart, again on the left and right side of the heart. The ventricle builds up enough pressure to send the blood out from the heart with a force we refer to as blood pressure. Take a look at the diagram for a better understanding. The blood that is pumped out from the heart typically makes this circuit through the following sequence of blood vessels. A large artery goes to smaller artery branches, and then arterioles, which are the smallest type of artery, a capillary bed, then to a venule, which is the smallest type of vein, to larger veins, and finally a large vein that takes the blood to the heart. Then to a venule, which is the smallest type of vein, to larger veins, and finally a large vein that takes the blood to the heart. The two sides of the heart form two major routes for the blood to flow along. The right side of the heart sends blood along a route that is called your pulmonary circulation. Along this route, the capillary beds are found in your lungs, where the blood picks up oxygen and releases carbon dioxide. The left side of the heart sends blood along a route that is called the systemic circulation. The artery that emerges from the heart at the beginning of this route is the aorta, which is located at the top of the heart. Branches of the aorta carry blood to almost every organ and every cell type in your body. It is a very important artery. Along this route, the capillary beds are found in your organs and tissues where the blood picks up your carbon dioxide and releases oxygen. Take a look at the diagram on the right for a better understanding. Here are some understandings that you should know of the control of heart rate.
The majority of the tissue that makes up the heart is cardiac muscle. Cardiac muscle spontaneously contracts and relaxes without any control by the nervous system. This is called myogenic muscle contraction. This activity does need to be controlled in order to make the timing of the contractions unified and useful. Within the right atrium, there is a mass of specialized tissue that has properties of both muscle and nervous system cells within its walls. This is called the sinoatrial node, or the SA node. The SA node acts as the pacemaker for the heart by sending out an electrical signal to initiate the contraction of both atria. For a person with a resting heart rate of 72 beats a minute, the signal from the SA node is sent out every 0.8 seconds.
The atrioventricular node, or the AV node. Within the right atrium is another mass of specialized muscle tissues. The AV node receives a signal from the SA node, delays for approximately 0.1 seconds, and then sends out another electrical signal. The second signal goes to the thick muscular ventricles and results in their contraction. This explains why both atria and then later both ventricles contract in synchrony.
During times of increased body activity, the heart rate needs to increase above the resting heart rate. This is because there is an increased demand for oxygen and cell respiration during periods of heavy exercise or activity. There is also a need to get rid of increased levels of carbon dioxide that accumulate in the bloodstream. As exercise begins and carbon dioxide levels begin to rise, an area of your brainstem called the medulla chemically senses the increase in carbon dioxide. The medulla then sends a signal through a cranial nerve called the cardiac nerve to increase the heart rate to an appropriate level. This signal is sent to the SA node and does not change the mechanism of how the heart beats, just the timing.
After exercise, the level of carbon dioxide in the bloodstream begins to decrease and another signal is sent from the medulla. This time the signal is carried by a different cranial nerve called the vagus nerve. Electrical signals from the vagus nerve result in the SA node once again adjusting the timing of the heart rate so that the heart returns to its myogenic or resting heart rate.
The heart rate can also be influenced by chemicals. One of the most common is epinephrine, also known as adrenaline. During periods of high stress or excitement, your adrenal gland secretes epinephrine into the bloodstream. Among other effects, epinephrine causes the SA node to fire more frequently than it does at its resting heart rate, and thus the heart increases, sometimes dramatically so.
Okay, so let's move on to the next understanding of how pressure changes in the left atrium, left ventricle, and aorta during the cardiac cycle. So, when both chambers are at rest, when the left atrium receives oxygenated blood from the pulmonary vein, both chambers, left atrium and left ventricle, are in the period of diastole, or when they're both relaxed. However, the pressure in the atrium, or the atrial pressure, is just slightly higher than the ventricular pressure. This results in the opening of the left atrioventricular valve, which is located in between the atrium and the ventricle. And with the help of gravity, the oxygenated blood is then allowed to flow through the valve into the left ventricle passively. And since the pressure in the aorta is so much higher than the left ventricles, as the pressure difference would keep the semilunar valve, or the valve between the left ventricle and the aorta, closed to prevent backflow into the ventricle. But what happens if the atria are in systole and the ventricles are in diastole? Well, since the walls of each atrium is relatively thin and is not really capable of creating very much pressure, and much of the volume of the blood has already been, transferred to the ventricle through the opened atrioventricular valve. Only small amount of pressure is needed in order to get rid of the rest of the blood that is still left in the atrium. So what about when the atria are in diastole and the ventricles are in systole? Well, as soon as the ventricular systole begins, the pressure inside the ventricle increases, so that the pressure inside the ventricle became greater than the pressure inside the atrium. As you can see on the picture on the right, in early ventricular systole, the pressure in the ventricle is at 15, while the pressure in the atrium is at 3. This then causes the atrioventricular valve to close in order to prevent backflow back into the atrium. And the pressure in the aorta is still much, much higher than the pressure in the ventricle, as you can see on the right, as the pressure in the aorta is at 75. So the semilunar valve, which is located in between the aorta and the ventricle, remains closed. However, since the ventricle is now filled with a lot of blood and is also very, very muscular, it allows the ventricular pressure to keep increasing as the systole continues. So then as you can see in the late ventricular systole picture diagram, the ventricle's pressure has increased from 15 to 125, which has then now become a lot greater than the pressure in the atrium and slightly higher than the pressure in the aorta, which then allows the semilunar valve to open, which then allows the ventricle to pump the blood into the aorta and the aorta to pump it to their body. And as the ventricle finishes the contraction, the pressure inside it drops and drops below the pressure in the aorta, and the semilunar valve then again closes, and this goes back over and over again, and this is what we call the cardiac cycle. So here's an overall picture of the pressures in the cardiac cycle. If you look at the first trend, which is the atrium trend, you can notice the little peaks that exist, which represents the small amount of pressure that is created in order to get rid of the remaining blood in the atrium that hasn't been passed onto the ventricle and passively passed onto the ventricle. And if you look at the ventricle trend, you can see that you can notice that the peaks are much higher, a lot higher at 120 millimeters mercury, as the ventricle is much more, it's a lot thicker, and it also contains a lot more blood. Therefore, it needs a lot of pressure to pump that to the rest of the body, to the aorta and to the rest of the body. And you can see that the aorta trend also follows the ventricle's trend, as the ventricular pressure is at its peak, the aortic pressure is also at its peak. This is because they have to work simultaneously. As the ventricle pumps the blood to the aorta has got to work it quick and then pump it to the rest of the body right away.
This slide's UIS is specifically about causes and consequences of occlusion of the coronary arteries. So basically, in the previous slides, we have learned that the coronary artery carries blood and oxygen from the heart to the different parts of the body, and it carries new oxygen. So basically, it feeds the rest of the body. And so if oxygen is deprived from the bloodstream, it will lead to an occlusion, which you could see from the slide. Occlusion is the blockage or closing of a blood vessel. Basically, when a coronary artery or one of its other main bloodstream branches become blocked, it is known as a coronary thrombosis, or, as I said earlier, a occlusion. So basically, what causes an occlusion? So occlusion is caused by atherosclerosis, or in other words, the buildup of plaque. As you can see from the picture on the right-hand side, the yellow part in the artery is the plaque, and it is in the endothelin, so basically the inner part of the arteries. The plaque is built up of lipids, cholesterol, cell debris, and calcium. Basically,
Plaque comes from all the things you eat. So basically, if you eat junk food or like sweet food, it will create plaque in your bloodstream, and it can also be caused by genetics. This plaque buildup can cause in over like your lifetime. So basically, it takes a long time for it to block your arteries, and the consequences of plaque buildup would make the arteries harder and less flexible. And so the consequences of occlusion of the coronary arteries would lead to parts of the body not getting enough blood, and it would also lead to a heart attack.
Moving on to the next understanding, William Harvey discovery of the circulation of the blood with the heart acting as a pump. Prior to his work, the authority of the movement of blood was given by the early Greeks. This was AD 100 to 200, and they believed that the blood was constantly being used up within the body, and they did not consider the closed circulation system that we know today. Galen taught his students a couple of things. He taught them that there are two kinds of blood: the nutritive blood, which was made by the liver, and the vital blood, which was made by the heart and distributed through the arteries to carry the vital spirits. He also taught them that blood traveled from one ventricle of the heart to the other through tiny pores, and that blood would seep from one location to another until the blood was used up. This was believed in medicine for over 1300 years. Galen's study led to a medical procedure known as bloodletting. So this was when small cuts were made in order to drain blood from certain areas of the body to balance the blood with other bodily fluids. And it was believed to restore the healthy balance. Furthermore, Harvey conducted years of animal dissections, live animal experimentations, and human cadaver dissections. His experimental work showed how the blood circulated around the body. He determined that the heart worked as a double pump, systemic and pulmonary circulations. And he determined that the blood is continuously circulated to and from the lungs and to and from the body. Although he was not able to see the capillaries that linked the arteries and the veins, he suggested their existence. And lastly, in 1628, Harvey published his work in a publication called On the Movement of the Heart and Blood in Animals. Many did not believe his findings, but the nature of science sometimes dictates that good, new scientific knowledge takes time to become trusted.